talk about two major nucleic acids. The first is going to be DNA, and the second is RNA. The first is, again, DNA, and DNA stands for deoxyribonucleic acid. Now, our DNA structure, as you can see from this picture here, consists of a double helix. It looks like a spiral staircase, right? So it's a very big structure. And the DNA contains all the genetic information that we need to make proteins. It's got all of our gene sequences so that we make different types of proteins, and it essentially makes us who we are. Now, DNA is composed of what we call nucleotides. Nucleotides are the building blocks of DNA. And each nucleotide is composed of a sugar, a phosphate group, and nitrogenous bases. Now, the sugar in DNA in particular is called deoxyribose. And then the nitrogenous bases are these right here. You have adenine, you have guanine, you have cytosine, and you have thymine. Now, keep in mind, again, this is a double helix. Now, when we compare it to RNA, RNA stands for ribonucleic acid. Now, ribonucleic acid is a single-stranded molecule. You can see it's just one strand, and it's made of, the nitrogenous bases are going to be adenine, guanine, cytosine, and uracil. So as you can see, thymine here in DNA is replaced by uracil in RNA. So let's talk about how these nitrogenous bases are paired up. Now, adenine always pairs up with thymine when we're talking about DNA. So adenine always pairs up with thymine. And guanine is always going to pair up with cytosine. And this concept is called the concept of complementary base pairing, right? If you have one strand on one side, you can predict what the other strand is going to be like if you know which nitrogenous base pairs up with which. So again, we know in DNA, in the DNA structure, we know that adenine pairs up with thymine. And we know that guanine pairs up with cytosine. Now, when it comes to the RNA structure, it changes a little bit. There's a slight change. Now, adenine in RNA is always going to pair up with uracil, not thymine, but uracil. And then guanine always pairs up with cytosine. Okay, so just to recap here, when we're talking about a DNA structure, adenine is always going to pair up with thymine, and cytosine always pairs up with guanine. Now, I picked up a little mnemonic device or acronym to kind of remember this. So I remember A goes with T, the golf course. So G for golf and C for course. So in DNA, you remember A goes with T, and C goes with G. Now, again, in the RNA structure, you have adenine pairing up with uracil and guanine pairing up with cytosine. Once you know how they're paired up, you can predict the sequence of either a DNA molecule or an RNA molecule. Now, RNA is important because it is a smaller molecule and it's able to move beyond the nucleus. And this is going to be important when we discuss transcription and translation, which are two important steps in protein synthesis. But that's going to be a topic for another video. This one, I just wanted to make sure you guys understood how these nitrogenous bases are paired up in the different structures, either in DNA or RNA. I hope this clarifies things up for you guys. And this concludes this particular video. Thank you so much for watching.